All right, this is a great chapter. I love molecular biology, molecular diagnostics. It is the future of the laboratory. So we're going to talk about serological and molecular detection of viral infections. And I'm changing things up just a little bit again. You have guided notes again, but this time the words are not on the screen. You're going to have to listen, and I will try to get the subtitles to work. All right, so beginning off, what is a virus? It is a non-living submicroscopic particle. It contains nucleic acid, DNA, or RNA, but not both, with inside a protein coat. Viruses are obligate intracellular pathogens, and they take over the host cell to replicate and proliferate. So how does the body fight viral infections? The innate immunity, that is the NK or natural killer cells, and cytokines like interferons. And if that fails, we have the second humoral cell-mediated defenses, or antibodies. IgA neutralizes viruses in respiratory and the GI tract. IgM is made first. It activates complement and inactivates virus by agglutinating particles. IgG activates complement, promotes phagocytosis, and we need to note that IgA is found in mucous membranes. IgG and IgM are in the bloodstream to inactivate and disseminate. IgM, if IgM proteins are positive, that means the patients have a current or recent infection. If IgG is positive, that means the patient has a current or past infection and immunity. So how do the viruses fight back? Viruses rapidly divide and mutate. They can evade the immune system. They can suppress the immune system, and examples of this are CMV, cytomegalovirus, HIV, human immunodeficiency virus, and EBV, the Epstein-Barr virus. And viruses can remain in a latent state waiting for the decline of the immune system due to other illnesses or old age. So how do we test for viral infection in the clinical lab? There are two main ways. Number one is serology test. They monitor the course of infection. Is this getting better or worse? Is it a current or past infection? That's what serology tests tell us. Or what's the patient's immune status? An example is if a pregnant woman, we need to know if she's immune to rubella. And how do we know? So the rubella IgG antibody will be positive if she is immune. If she is negative for rubella IgG, that means her baby is at risk for infection because she is not immune. The second test group that we do are molecular tests. They detect current infections and tell us if antiviral therapy is working. Molecular tests detect the number of viral particles per milliliter of blood. It is just amazing. So some examples of that would be hepatitis C RNA. If the patients have less than 100 copies per milliliter, that means if they have less than 100 copies of the RNA for hepatitis C in every milliliter of their blood, it will still be not detected or negative. If the RNA copies are 200,000 to 1 million, that means there's a low infection. A 1 million to 5 million copies of RNA per milliliter of blood is medium. 5 to 25 million would be high, and greater than 25 million would be very high. So you can tell, you can do a quantitative or viral load testing to tell exactly how many viral particles are in each milliliter of blood. It is just amazing. So the next thing we're going to talk about is molecular tests. What are they and how do they work? and we are jumping to the molecular diagnostics chapter. So we're kind of squishing together the viruses and molecular. So the first test method that we're going to talk about are hybridization assays, or quote unquote blots. So chemically, you crack open your target cell and isolate the nucleic acid or protein of choice. Like if you're looking for DNA, you would extract the DNA. If it's an RNA virus, you would extract the RNA and protein, so on. You would stick them to a type of support like a glass slide, some kind of gel or filter paper. A probe is then mixed and added to, or mixed around to bind. And a probe, this is on your study guide, is a complementary nucleic acid sequence specific to the target you're looking for. A dye or radioactive label is added so we can see a positive or negative reaction. And a blot is gel electrophoresis followed by transfer of the pattern to a membrane, like a nitrocellulose membrane or something. And there are only 3 kinds of blots on your study guide, but there are actually 4, and we won't go into the 4th one. I'll just mention it briefly. So the first one is the Western blot. In the Western blot, they are looking for or probing for protein. The Southern blot is looking for DNA, and the Northern blot is looking for RNA. And the 4th one that we don't really care too much about, the Eastern blot, just know that there is one. So I guess somebody was being funny. Then the next method that we're going to talk about is the DNA chip technology, or microarrays. In the microarrays, DNA or RNA probes are anchored to a surface, usually a glass slide. So if you look at this glass slide in this picture, you see these frosted, opaque squares. If you look at this, you can tell that everywhere there's a dot, there is a complementary sequence, like a probe. DNA or RNA can be placed on the slide. So everywhere these dots are, that's either DNA or RNA, and there can be like tens of thousands on one glass slide. It's just amazing. So usually we start with 1 DNA or RNA probes anchored to a surface, usually the glass slide. Then 2 isolated RNA or DNA from your patient that you extracted is added and allowed to bind or hybridize. That means the complementary sequences connect together and bond. Then number 3, fluorescent tags are added so we can visualize our target. And then you can test up to 10 to 20, up to hundreds of thousands of nucleic acid sequences at one time. So let's try a video. So with DNA microarrays, each dot represents a sequence, a known sequence. You add tagged samples of DNA, and everywhere that there's binding, you see fluorescence. Then computers are used to read the chip and analyze it. So you can do gene expression profiling, protein microarrays. So for gene expression profiling, for example, in the liver, you could take some liver tissue, extract and isolate messenger RNAs. So messenger RNAs that means the protein is actually being expressed. You can produce complementary DNA. Then you can do a whole genome chip where the whole genome, complementary sequences are on the chip. Then you add a selection of those tagged DNAs that you've made, and you can identify which genes are being expressed in this sample by noting which DNA fragments are positive. It is so cool. For protein microarrays, you have like an antibody microarrays to specific proteins are attached to the slide. Then you add the proteins that you've extracted that are labeled, and those bind to those specific proteins to define which proteins work together. bound proteins. Dots on the chip show positive only where protein are bound. And then comparative genomic hybridization. This is pretty cool too. You have the whole genome of some species. Then you chop up a mixture of the whole genome of a test and control sample. And then you can dump it on there together. And it's competitive testing. So you get this nice, you can detect major deletions and chromosomal abnormalities. It is pretty amazing. And this is where we get into cancer genomics, and we can tell genes affected that cause mental retardation. It's so cool. So we're going to recap that video on your study guide. So this is, we're going to start with the 3 types of microarrays. And they are gene expression profiling. That tells us what genes are actively being expressed because we've extracted mRNA or messenger RNA. DNA sequences that are actively being made into proteins can be found by looking for the second step in DNA to protein cycle where a messenger RNA copy is made. Then number 2, we can have protein microarrays where we are looking for using proteins or antibodies. Then number 3 is CGH, or comparative genomic hybridization, or whole genome DNA. The purpose of microarrays is to identify unknown nucleic acids and compare healthy and diseased genes. Now we're going to talk about the problems with hybridization or chip technology. Number 1, non-specific binding can occur. A, we can get primer dimers. B, we can get binding of the wrong patient strands with probes, or non-specific. That's non-specific. C, we can actually get patient-to-patient binding. 2, test conditions vary. A, salt/buffer concentrations. B, temperatures. C, humidity. For example, Psi-3 or Psi-5 won't work in the Mississippi summer. 3, the target sample, either DNA or RNA, whatever you're looking for, is in too low of a concentration to detect. The next thing we're going to talk about is DNA sequencing, and there's not a picture here, but DNA sequencing is when we use a machine, and that's used to commonly detect deletions, mostly. Then next we have target amplification, and these target amplifications include PCR and real-time PCR. PCR, or polymerase chain reaction, is done by first you have to extract DNA or RNA, and it will be RT or reverse transcriptase PCR if it's RNA. And you have to make a cDNA copy. So you either have to start with DNA or make a complementary DNA copy of your RNA. And you do that by using a primer set, both forward and reverse, because if you remember back to general biology, DNA and RNA has a 3' end to a 5' end, and they are complementary, so the opposite end will be opposite. So the 3' matches with the 5' of the other end, and vice versa. You need a master mix, and the master mix is free nucleotides with magnesium, buffer, EDTA, and enzymes. And yes, it's the same EDTA that's in our purple tops. You load the thermal cycler and set the temperatures and times and number of cycles, usually 35 cycles, and wait about 2 hours. So this is a picture of real-time PCR. We're going to come back to this in a second. All right, so how does PCR work? First, you have to denature your double-stranded DNA at 95 degrees Celsius for about 30 seconds. Number 2, you cool to 37 degrees C to allow primers to attach. This takes about 15 seconds. Number 3, you heat the sample to 72 degrees C so the Taq polymerase can anneal, make a complementary strand, and that takes maybe 45 seconds. So Taq polymerase is an enzyme found in Thermus aquaticus. It's a bacteria found in hot springs and the geysers at Yellowstone, and it can make a copy of DNA, Taq polymerase. So after those 3 cycles, and that's considered a cycle, the heating to 95, cooling to 37, and heating back up to 72, that's one cycle. Now you have 2 copies, and that's represented by this picture right here. So we heated it to denature it. We let the primers anneal, and then we did the extension. So now we have 2 copies, and we can do the same thing again. Now, you repeat this process, the 95 degrees Celsius, 37 degrees Celsius, 72 degrees Celsius, 35 times or so. Real-time PCR works in the same principle, but we use a fluorescent tag and take digital pictures so you can watch your copy number grow in each time. So after every cycle, the instrument takes a picture, and you can see this fluorescent tag will only stick to double-stranded DNA, so your completed copies. That's how you know. So Roche, R-O-C-H-E, has the patent, so it's very expensive to use the PCR technology. There are now several variations. The drawbacks of molecular testing: false positives come from contamination, number 1. False positives come from contamination. 2, the need for clean rooms or separate areas for extraction, amplification, and detection. And 3, more QC is needed. The advantages of molecular testing is that it is more specific and more rapid. Okay, so now we're going to get back to the viruses. So hepatitis A, or HAV, is an RNA virus. It is transmitted by the fecal-oral route from person to person through ingestion of contaminated food or water due to poor sanitation, hygiene, and overcrowding. The incubation period is 28 days, and it usually resolves on its own in approximately 2 months. There is no chronic state, and it is self-limiting. It is diagnosed by the presence of IgM antibodies to hepatitis A virus by EIA. So anti-HAV IgG persists for life post-infection. So that's what gives you immunity, the IgG that lasts the rest of your life. So you have a chart on your study guide, and for acute infection, what goes in the first box is anti-HAV IgM. In acute infection, the IgM is detected, but in past infections, IgM antibodies are not detected. An anti-hepatitis A virus IgG is not detected in acute infection, but it is detected in past infection. So these charts, these boxes are very important. You need to remember that IgM means current or acute infection, and IgG means current or past infection. The next virus we're going to talk about is hepatitis E virus. It's an RNA virus. It's transmitted by the fecal-oral route from person to person from ingestion of contaminated drinking water. And I put this picture of drinking water right here to jog your memory. The incubation time is 3 to 8 weeks, and it resolves in about 1 to 4 weeks. There is no chronic state. It is self-limiting, and it is diagnosed by the presence of IgM to HEV by EIA, and so that would be anti-HEV IgM. That would be an acute infection. Mortality rate is increased in pregnant women. And so you can see how the liver enzymes rise. So this would be your ALT. It's a liver enzyme that's rising with hepatitis E. This is the IgM, the yellow. See, it starts first. And the IgG starts about the same time, but it takes a little longer to get up to detectable levels. Hepatitis B is the DNA virus. It is the only hepatitis virus that is a DNA virus. It is transmitted by sharing needles, sexual contact, contact with contaminated blood or body fluids. Examples are blood transfusions, tattooing, drug users, and needle sticks. The incubation is 45 to 90 days. It resolves in 6 months to never. There can be an asymptomatic chronic carrier. For those that have symptoms, they can last weeks to months. Most adults do recover in 6 months and develop immunity. 1% of develop disease with necrotic hepatitis, and so this would be like an example of necrotic hepatitis, this poor person here. The lab has a very important role in diagnosing, staging, and following treatment of hepatitis B. Next, we're going to talk about the tests for hepatitis B. There are many, and it is very confusing. Oftentimes, we have to help people decide which hepatitis test they want to order. Hepatitis B surface antigen, or HBs antigen, a protein that's found in the outer envelope of the virus. It's the first marker to appear at 2 to 10 weeks post-exposure and become undetectable by 4 to 6 months after symptoms. The hepatitis B surface antigen, if it is detected or positive, that means you have an active infection. So the antigen present always means active infection. The hepatitis B E antigen is a protein found between the surface and the core. So if you notice, this is a picture of the viral particle of hepatitis. You have the outer surface, the outer inner envelope. It's a very complex virus. So hepatitis B E antigen, it's a protein found between the surface and core. It's the second marker to appear shortly after hepatitis B surface antigen. It does not stay elevated long, and it can be positive in chronic infections. And increased levels mean the virus is actively replicating. So that's interesting that the hepatitis B E antigen, even though it's an antigen, it can still be positive in chronic infections. The thing to note here is that the hepatitis B core antigen, or HBC antigen, is not detectable in serum because the envelope masks it. That's why we don't test for it. Hepatitis B core antibody, usually we do a hepatitis B core total, which is the IgM plus the IgG. Then we have the anti-hepatitis B core IgM, which is the first antibody to appear after infection with hepatitis B. Then the anti-hepatitis B core IgG production overlaps the anti-hepatitis B core IgM. Then number 5, we have the hepatitis B envelope antibody. This indicates recovery. And we have the hepatitis B surface antibody. This indicates recovery and provides immunity. Testing methods for hepatitis include commercial, automated immunoassays, and molecular tests. Real-time PCR is the most sensitive methodology, detecting as few as 10 copies of hepatitis B DNA per ml of blood. Isn't that amazing? It's just amazing. For hepatitis D, I don't have a picture, but hepatitis D is an RNA virus. It stands for hepatitis delta, and it can only occur with co-infection of, oh, I do have a picture. I'm so sorry. All right, so hepatitis C is pretty famous. Oh, I'm sorry, I've all messed up. Okay, hepatitis D, an RNA virus. Hepatitis delta can only occur with co-infection of Hep B because Hep B carries the enzyme required for Hep D to replicate. It is most common in IV drug users, and a chronic state does exist. Now, hepatitis C is an RNA virus. The transmission can be from contaminated blood and body fluids, through IV drug use, blood transfusions, and organ transplant pre-1992, because 1992 is when we started testing for hepatitis C in the blood bank. Incubation is about 7 weeks. It varies from 2 to 30 weeks. Most infections are asymptomatic, but 85% develop chronic cirrhosis. Chronic cirrhosis from Hep C develops very slowly over 20 to 25 years and carries an increased risk of liver cancer. Hepatitis C is tested with serology and molecular pathology. The anti-hepatitis C virus IgM means acute, recent, current infection. So acute/recent/current infection, any of those for IgM. The anti-hepatitis C virus IgG means past infection / immunity. And these are some famous people that have hepatitis C. Maybe it should be a bonus point to name these people. And here is a nice chart that I made. You definitely need to remember this stuff. And we will talk about this in lab because it is very, very confusing. Next, we have the EBV, or Epstein-Barr virus. There are several different causes. This causes several different kinds of disease. We have lymphoproliferative diseases, cancers, malignancies, and infectious mononucleosis. By adulthood, over 90% have been infected. The transmission is for intimate contact with salivary secretions from an infected individual, also known as the kissing disease. It can be transmitted, less often seen, by blood transfusions and organ transplants. So it's not common, but we can see it. What happens is the epithelial cells of the oropharynx, or throat, are infected, and it looks terrible. Viral particles infect B lymphs, which spread the virus through the lymph system. And the classic symptoms include fever, lymphadenopathy, sore throat, and last 2 to 4 months. Fatigue can last for months, and severe cases include liver and spleen enlargement. So what tests do we do in the lab? We do a CBC, where we will see absolute lymphocytosis with greater than 50% lymphs and at least 10% atypical lymphs. So these are atypical lymphs right here. They kind of look like monos, but they are not monocytes. They are reactive or atypical lymphs. Now, in serology, we can do the heterophile antibody. Heterophile antibodies are antibodies that are capable of reacting with similar antigens from 2 or more unrelated species. The heterophile antibodies associated with infectious mono, or IgM antibodies, capable of reacting with horse, sheep, or bovine, that means cow, red blood cells. Also known as the Forsman antibody. F-O-R-S-S-M-A-N. 40% of patients infected with infectious mono produce these heterophile antibodies during the first week of illness, and 90% make them by the fourth week. The heterophile antibody, or monospot, was done as a screening test for infectious mono. We don't really do this anymore. The monospot tested the ability of the patient's serum adsorbed with guinea pig kidney or beef red blood cell antigens to agglutinate horse red blood cells. The antibody could then be titered by incubating serial dilutions of the patient's serum with sheep red blood cells in the Paul-Bunnell test. Now, this is mostly trivia information. These methods have been replaced with rapid latex agglutination and immunoassays. We should note that adsorbed, that means the antibodies in the serum are sticking to the antigens on the guinea pig kidney and beef red blood cells. Paul and Bunnell created this method, and it was named after the two American physicians who devised this test in 1932. So the way this heterophile test works is that it relies on the agglutination of the horse red blood cells by heterophile antibodies in patient's serum. A 20% suspension of horse red blood cells is used in an isotonic 3 to 8% sodium citrate formula. So we could go on, but anyway, just know that what the heterophile antibody is, and that Paul-Bunnell was one of the first tests for infectious mono. Okay. There are also antibodies to certain EBV antigens. We have the anti-VCA IgM. VCA stands for viral capsid antigen, which is part of the EBV particle, and if it's positive, it would indicate a current infection. We also have the anti-VCA IgG that can also be present in current infections, but will persist for life. Other markers of EBV are the EBV-EA, which stands for early antigen, and EBV-MA, which stands for membrane antigen. We also have the CMV, cytomegalovirus. It is ubiquitous, means that it is everyone. Nearly everyone is exposed through close contact. It's not a big deal unless patients are immunocompromised, like pregnant women, infants, elderly, and patients on immunosuppressive therapy. So if you have a CMV IgM that's positive and CMV IgG that's negative, that means you have a recent infection. CMV IgM, if that's negative and the IgG is positive, that means you're recovering from infection. CMV IgM negative and CMV IgG negative means that you were never infected. And the very last virus that we will talk about is VZV, or varicella zoster, that causes chickenpox and shingles. VZV IgM, if that's positive and the IgG is negative, that means there's been a recent infection. And if the IgM is negative and IgG is positive for varicella, then that means you have a past infection or you have been vaccinated and you have immunity. And that concludes our serological and molecular detection of viral infections.